Hello, today I'd like to talk to you and introduce you to scientific literature, and I'd love to show you the parts of a lab communication and discuss how to write a lab communication for Biology 159. Today's objectives will be to discuss the parts of a lab communication. So what is a lab communication and what are the parts that we usually have in a lab communication? We're going to compare that specifically to a scientific paper, generally written in APA style. Scientific papers will have abstract, introduction, materials and methods, results, discussion, and references. I'm also going to mention to you how you might use the references sort of as in a backwards fashion to go backwards and find papers of interest. We will look at different databases, specifically UT Library databases, as well as Google Scholar, and we might practice how changing search terms will give us different results when we're doing literature searches. We will then go through a good versus bad lab communication example. I'd love to show you the rubric and checklist. These are some resource documents that we have for you, and then I'll wrap it up by assigning you to write a draft lab communication for the antibiotic resistance lab. Don't panic about this assignment though. This is only a draft. You're going to bring this thing in a couple of copies. You're going to pass it around to some of your peers in the lab, and they're going to give you feedback. You're going to get a chance to take that feedback home and then write up your lab communication before you turn it in. All right, so what types of scientific papers are there? There's a variety of types, and one that we have read so far are popular science papers. So when you read the What's So Hot About Chili Peppers by Burrell, that is a popular science paper. When you read the paper on antibiotic resistance, the new tactics for tuberculosis, that is also kind of a popular science paper. There's also primary literature, which I'm going to show you, and review papers, which I'll try to show you as well. These are both very good, but they're different than popular science. All right, so what is a popular science paper? Where is that? Let's see. Where's my two? Here's What's So Hot About Chili Peppers. Here we go with it. Let's go back to the beginning. So what makes this popular science? Well, this is written in a form that anybody can pick this up and just read it away. So it's written for laymen. It's also written by Burrell. So the article is Burrell, but it's written about the work of Joshua Tewksbury. So we are reading about the work of Tewksbury written by Burrell. So oftentimes reviews, or popular science type papers, you've got one author writing about the work of someone else. They're not writing about their own work, okay? In addition to that, it's kind of like a storyline to this paper, so it grabs the interest of any reader. There's lots of lovely pictures that kind of hook interest. And it's easy to read. There's no charts and data, like there's no raw data in here, okay? Still a very, very useful paper though, right? Another one that you've probably read a little bit on, hopefully, is this. This is written by Berry and Chung. This is New Tactics Against Tuberculosis. This is also a review paper. You'll notice here that there's no charts and tables. It is a storyline. There's lots of pictures, grabs the interest. This one's a little bit more sciencey. There's a little bit more jargon and things like that, but it is still a popular science type paper. It's very, very informative though, okay? Let me scroll down to the very, very end of this paper. Yeah, there's no real references list. Of course, it does, list out its sources. It tells you what the sources are, but nothing like that. Okay, now what about primary literature? A paper coming up that you're about to read, written by Slater et al. So here's G.J. Slater and the other authors. This one, you'll notice, is very different, okay? There's not a lot of really nice pictures. There are some images, don't get me wrong, and there can be images in primary literature, but the images shown in primary literature are going to be data, all right? You're going to have also the parts. Primary literature is going to contain an abstract. This is basically a summary paragraph that tells you a very streamlined summary of what this paper is going to show. They give an introduction, which provides background information. The introduction generally is going to provide background information that leads into the storyline and the research question of the paper. All right, so it's not just random background information. This is actually specific and important background information that tells the reader what we know specifically about this exact subject. It generally will then include the hypothesis and prediction, if there is one, of a paper. You're going to see that Slater doesn't explicitly state their hypothesis and prediction, but if it does, that's where this would be. And also the research question of the authors can be there. You'll see the methods. This is going to be a description that provides details on how the experiment was done. Step-by-step protocols, information about statistics. This should be detailed enough that someone could pick this up and repeat the procedure. So here we're continuing on with that. Then we finally get into results. Results section is going to include images. These are visual results as well as written results. Written results are just, there's no interpretation kind of here, no analysis of the data at this point, really just a visual with your, charts, tables, graphs, whatever kind of data you're including, and then a visual description, or a written description of what this chart, graph, table, etc. Is showing. All right. Finally, then they're continuing to show their data here. You move on to the discussion section. This is where the data analysis occurs. This is where we interpret the data and we say, what does this data mean? We will talk about what's known and if the data will fit in with what's known versus if the data does not fit in with what's known. That's okay. It just might be telling us something new. We talk about what questions remain, what type of future investigations should be done. We talk about any weaknesses in the data, errors, things like that. Then finally, there's a references section. This is where we include all of the references, the citations, the information of the papers that we included information from. So in a paper, you will cite. You're going to do this. You're going to use, this is APA style. You list out the author, then the year, title of the paper, the journal which it was in, then volume and page range. That is sort of the style, and it's in hanging indentation format. You see that in the references section. In the actual text, you would see an in-text citation like this, in parentheses, so at the end of a sentence, for example, large forces are likely to be generated by moderately sized prey during prey apprehension. This information came from this paper. So they included an in-text citation here. Prushoft and Witzel, 2005, so it's author name and year. And then, of course, over here, you would have the full information. We're going to have to go down because it's P's. Here we go. Here's Prushoft and Witzel. Here's the full information for that paper. So that's what a primary literature paper sort of looks like. All right. A review is a little bit different than that. A review will include information from several primary literature papers usually. It's kind of a collection of information. They're reporting on the results of a collection of studies. All right, let's go back to this now. So what are the parts of a scientific paper? We just looked at them. The abstract, introduction, materials and methods, results, discussion, references, or citations. Written lab communications, interestingly, are going to have the same parts, minus the abstract. You guys won't make an abstract. You will use common format. We'd like it to be 1.5 to 2 pages long. That does not include your tables or figures. Those can be additional. It should be single-spaced, 11 or 12-point font. All right. You must include a title. We'd like that centered at the top. It should be descriptive. All right. It should say something and tell the reader something about the investigation being done. An example of this for plant fungal could be how capsaicin affects fungal growth. Not lab 1 or lab X. Something like that that's not descriptive, okay? This is accepted, this is not. You're going to have an introduction. In this introduction, you're going to provide background information, right? This paragraph relates to your investigation to what is currently known. We would like it if you would at least cite the lab manual, the section, that you read on that topic, whatever we're doing at that point. And then you need to cite the paper that we have assigned you to read. And usually you will cite one additional paper. All right. When you cite this information, right, you are summarizing in your own words what is known. All right. For plant fungal, we didn't. We've given them sort of the lab manual stuff and Burrell paper. We also gave the initial observation data, right? You guys looked at Bell 1 pepper growth on C0, C1, and C4 plates, so you could include the information from those paper resources and lab manual resources as well as initial observations, and you should do that for this paper. And then you will cite in text the sources that you use as well as at the end on the references page. All right, in addition to that, in the introduction, you're going to lead in to your research question and then your group's hypothesis and prediction. You should say why you think this will be the result based on information you've presented in that background. Your introduction and summary is going to be 2 paragraphs in length. All right, you're going to include background information that leads into your research question and into your group's hypothesis and prediction. So in addition to the content, we are going to be looking at these 2 paragraphs for coherency. Quality of content. It should be relevant, not random, unrelated facts. And then the background is going to be leading in, like I said, to your research question, and it should frame your hypothesis and prediction. Next, you're going to have a section on materials and methods. This should include your experimental design. So you're going to have an outline of your experimental design, talking about variables, dependent and independent, any controls, any replicates, what are the steps of the protocol, provide enough information that someone else could pick this thing up and replicate the experiment. You're going to then talk about data collection. You should describe how you collect your data, what tools you use, then describe how you analyze your data, right? You guys are collecting data, you're calculating means, you're calculating standard deviations, standard errors, you're doing t-tests. We should talk about that and say that we've done all of this in Excel. This should be in paragraph form, not a list. Then you're going to have a results section. You should have both a visual representation section of results. This is not in the 2-page limit. It's kind of attached to the back of your lab communication, and as a minimum should include 1 figure. A line or bar graph, generally a bar graph, but sometimes line is more appropriate. But you're going to have 1 figure with appropriate labels and error bars. You may also include a table of your raw data if you like. Some TAs like that, some don't require it, some do. Make sure you find out. But this does not replace or count for the bar graph, okay? And then you can have a written description of your results. This is included in the 2-page limit, and it's sort of paragraph form, written description of what you see. It should reference the figures, right? So if you've got a bar graph, this is your figure 1, you should say, in figure 1, we see that blah, 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 all right? And then you could describe what you see and end the sentence with figure 1 instead of doing this format if you like. You should state what treatments were used, if they were different from each other, and how you know, a p-value. Nothing about what it means, it's just kind of a statement here. We use this treatment, light versus dark. This was statistically different. We know because the p-value was blah, blah, blah. All right, then in the discussion, this is when you want to say something about your data, more about what it means, right? This is where you can explain your data. This is where you can say, does it fit with what we know? Does it not? Does it fit into the big picture? Do they make sense based on what is known? How does the study add to what is known? If it doesn't fit into what is known, that is okay, but how could you explain it? Are there alternative explanations? Are there weaknesses, sources of error? Why does the audience care about the results? What questions remain? What future investigations can be done? You should also state whether or not you were able to support or reject your hypothesis. Don't say your hypothesis is proven. You could say the hypothesis is supported, or we have to reject it, right? In science, we never say anything is proven. Just be careful there. Talk about how your data and stats lead to your conclusion. Make your claim here, and then refer back to your data. You will also have a references cited section, APA format. Make sure you are citing the lab manual, the assigned paper, and one additional article. Please do a spell check as well. We will be checking for spelling and grammar, 1 point maximum. You will upload your document onto, when you finally turn it in. You don't do this for the draft yet, but you will with your final copy, upload it to Canvas into the plagiarism checking software. Because we are going to write our lab communications individually. We are not going to copy from the group oral communication, right? No, we are not. We're going to put this in our own words. You are expected to do this individually. Literature searches now, right? Because I told you I would show you how to do this, because in some papers, you know, you're going to have to learn how to find your own papers. We have assigned papers, but you may go to Google Scholar or the UT Library databases. Let's do that now. For the UT Library databases, I went to library.utk.edu right here. I love this because from here you can also go to Google Scholar if you like, whatever you want to do. I like to go here to the articles and databases link. You can then search by science, or excuse me, by topic. You could also search by type. You can also search by popular databases. Some really good ones are Web of Science and PubMed. Let's go to Web of Science. I really like that. It is going to ask you to log in with UTK user and password. Let's don't remember that, please. Let's just go on. I'm logged in now. For example, if we were searching capsaicin fungal growth, let's search for that. We can narrow down and select for just certain year ranges. You can do certain authors. You can look for certain types like primary versus review if you like. Let's just do a random search here. Look at what I'm finding. Ooh, capsaicin as a natural preservative against fungal attack on Pinus species and Hymenaea species. Ooh, this is an Aspergillus. That is a type of fungus. Ooh, they're using red pepper, powdered red pepper, to affect the infection of this type of fungus. Ooh. Red hot pepper agar in the isolation of Candida yeast. Nice. All right, so we've got lots of information here. I like this one. Let's look at this one. This is a primary article. I know this because I see the abstract there. If I want the full text, I'm going to click on this link, and I'm going to grab that full text. I can also find, let's go back, excuse me. Click back. All right. What if we change our search terms, right? What if we change our search terms? Let's go back to our original search and add review to it. Ooh, we found nothing, so that wasn't very useful, was it? Let's do a structure search. Oh no, it's wanting me to sign back in again. Let's don't do that. Let's don't do that. Let's say capsaicin effect on fungi. Let's see if we find the same papers. We will probably find different papers. Shifts in plant chemical defenses of chili pepper. But look at these. These are general papers. Are these helpful for ours? What if we did What if we did light versus dark? And none of those papers are really helpful for us. Ooh, here's some information on light-dependent development of fungi, daylight exposure, and bacterial communities. Ooh, so it's talking about light exposure on bacteria. That's interesting. It's not really fungus, but you could relate that. Let's go back. Let's see if chili peppers light dark gives us. Ooh, this is great. Seed germination of wild chili peppers. Light versus dark. All right, so do you see how I went from fungal growth and capsaicin, which is what many of you guys would look for, to something more specific? If I'm doing a light versus dark, what if we're doing temperature? Would it be helpful to look at chili peppers? Oh. This is talking about drought. Ooh, here is a temperature and a capsaicin effect on humans. That would be really cool. Cold treatment, chili peppers. So, general search terms give you general results. Specific search terms give you more specific results, right? Nice. All right. Now, how are you guys going to use this information? You guys are going to use this information to write a draft lab communication for the antibiotic resistance lab. All right, you guys are supposed to be using documents posted on Canvas. You're thinking, where are those? I'm going to show you those. And then you're going to use them and write this up. And you're going to print 2 copies to turn at the start of next class. All right, so where are our documents? Let me show you the documents we have got for you. Here we go. Here is a description document. Just updated. This document describes the purpose of a written lab communication, describes format, goes through in each section and gives you the point values and what you must do. Here's the methods, results, visual and written. Here's your discussion, talking about your hypothesis, analysis of your data, implications, future investigations, references, all of the components. Okay, there's that document. That is posted for you. You can read it. Please read it. Here is a communication checklist. You can almost, this is posted for you as well. You can almost use this as your outline. I would almost pull up this document, save it to your computer, right? And then start typing in your pieces and flesh this thing out. It is an outline. All right, you can use that document. We also have this for you. This is, I love this. This is using, let's see if that's too big. No, that's perfect. Let's stretch this out as big as we can get it, right? This is going to be using, and it's using the plant fungal lab communication. It's an example using that as the topic. And it is a good versus bad, okay? So it goes through and shows you a title. It shows you a couple of bad titles. Fungal growth lab report. Needs more detail. Here's another bad one. This is just too long. Tests whether high amounts of capsaicin, C4, and high temperatures will result. And this is like a paragraph. A good one here is the effects of capsaicin and temperature on fungal growth. That's beautiful, right? It's descriptive, but not too long. Here's an introduction that is bad. This one is bad because there is direct copying and pasting happening here, so it's plagiarism. It's also bad because there is just kind of incoherent information. It's random fungal facts. And it doesn't have a nice flow. All right. There's also a problem here because there's information from a plate. There's information from right here, a paper, but there's no citation. So there needs to be an in-text citation there. Here we're looking at their hypothesis. There's an issue with this hypothesis in that the hypothesis is not testable. It should include an explanation and be more descriptive as well. Here, on the other hand, is an example of a good lab communication introduction. This one is using proper citations, proper, coherent, flowing information that leads right into their research question and their hypothesis. The hypothesis is testable. It's beautifully worded. Some tips. You may need to read lots of papers when you write the introduction. You can access most databases. Here's the UT library information. It also points you to look at Web of Science like I showed you. You can also search keywords using the author's names. And please don't forget to cite papers in the in-text citations and on the references page. Also looking at the discussion, since the discussion is often a difficult section, we're showing you an example of a bad discussion and an example of a good discussion. Okay. So here we want you to say whether the hypothesis was supported or not, supported or rejected. You don't want to say it was accepted or proven. We never say those words in science. Here there's an issue through the next error I see is that they talk about a significant difference there, but they don't state the p-value. So here the comment is telling you, where's the p-value? We need to know. You can't just say that it's significant or not without stating it. Here's another problem. They talk about there weren't any extreme outliers. How do they know? They need to point back to that standard deviation. Was the standard deviation large or not? If it's large, you can say, yeah, we had outliers. If it's a narrow standard deviation, small standard deviation, you probably didn't have any outliers. So you need to, again, point back to the numbers, right? Implications, talking about known information, effect of humidity and capsaicin, preventing fungal growth, you need a citation. Anytime you cite information, you need an in-text citation. There's a problem here in future investigations. They are basically using a wider temperature range, and so they're repeating the experiment. There needs to be something unique done there. They've just added in a couple more temperatures. Whereas if you have a good discussion, here we're going to be talking about that information and then citing the p-value. We're going to be talking about outliers, and we're going to say that because we're going to talk about the standard deviations. When we talk about future investigations, we're going to have something actually unique. We're going to look at humidity instead of just temperature there. There's in-text citations everywhere appropriate. And the numbers are actually shown. In results, here's an issue with some results. They've shown just a picture of their results taken by a phone, right? We need to actually include the graph from Excel. You would want to copy and paste the graph from Excel into your document. Again, the table should be a table, not a picture. You need a table to have a title. We need to make sure that if there's an obvious difference in groups, we have to talk about the p-value, right? This is showing you a good figure. And it's got error bars, all right? Materials and methods here at the end, just in case you wanted to look at that as well. Here's some information on good and bad experimental design, right? Bad, it's too vague. They're not giving enough detail. They don't include the number of plates. Some dishes had C4 in them. How many, right? We don't know. Data collection, we don't know enough information about that. It should be written in paragraph form, not bullet listed. Here's an example of good experimental design with enough detail, enough plates, exactly the number, how long they were incubated, for example, written in paragraph form. References section, bad. We are not using APA in this example. Good, we are using APA. A couple of tips, if you want to, you can use EndNote tool. It can help you manage your references. It is free for UTK students. You can download it from the OIT website. And Google Scholar also will offer different citation styles. You can select that, and then when you search the paper by Google Scholar, you can copy it into your references section in the proper format. All right, so what will you all do? You all are going to take this video, you're going to take this good and bad example document, you're going to take the checklist and the rubric, the description document, and you all are going to give this a stab, right? You're going to write a draft lab communication for the antibiotic resistance lab. I want you to write a draft entire thing, write an intro, write a materials and methods, write a written results, visual results, and a discussion section. Also a references section. Don't forget to include in-text citations. All right? Do your best. A recommendation is to open up that checklist and use that sort of as an outline and start typing your stuff in and kind of putting things in piece by piece. Once you have your draft ready, you're going to print 2 copies and bring it to you to the start of the next class. One will be turned in and one will be used for getting feedback, right, from your peers. You will take that feedback then and you will have some time to incorporate that feedback before you actually write up the final version and turn in the final version in class and on Canvas into the unit check. All right. Please let your instructor know if you have any questions, and I hope you enjoyed this video. Thanks.